Welcome to today's webinar, a joint initiative between NATHEALTH and the American College of Healthcare Executives. Thank you for joining us today. I'm Molly Lowe, and I'm the Director of Content Strategy for the American College of Healthcare Executives, also known as ACHE. We are honored to work with NatHealth on this joint initiative and have the unique pleasure of having an international audience today, with attendees joining from all over the world. Before we get started, I would like to go over a few items so you know how to participate in today's event. Audio will be heard through your computer speakers, so please be sure that your volume is turned up. If for some reason you're having difficulty with the connection, our recommendation is that you exit out of your browser and restart the Zoom session. You will have the opportunity to submit questions to today's presenters by typing your questions into the Q&A box. You may send in your questions at any time during the presentation. We'll collect these and address them during the Q&A session at the end of today's presentation. The link to the handout for today's webinar can be found at the bottom of the reminder email you received from ACHE today. It was sent to everyone about an hour ago. Next slide, please. Today's event will offer each participant 1 hour of qualified education credit. Credits are self-reported for this webinar. To receive your credit, visit my.ache.org and select My Education Credit on the right-hand side of the screen. Scroll down to the self-reported credit section and simply add your credit. Today's program is being recorded and will be available after the webinar. Each of you will receive a thank you email tomorrow from ACHE containing a link to the webinar recording and the handout I previously mentioned. It will also include a link to an evaluation form for today's webinar. We greatly value your feedback and hope you'll take a few minutes to give us your input. Next slide, please. ACHE would like to take a minute to recognize all of our 2021 Premier Corporate Partners for their continued support of ACHE. These organizations play a vital role in supporting ACHE's vision and mission to advance our members in healthcare leadership excellence. You may find additional resources provided by our partners within the ACHE COVID-19 Resource Center and the Corporate Partner section of ache.org. Next slide. It's now my pleasure to introduce everyone to today's speakers. Next slide, thank you. Thank you. Hirsch Mahajan, MD, David S. Perlin, PhD, and K. Srinath Reddy, MD. Dr. Mahajan is the president of NatHealth and the founder and chief radiologist of Mahajan Imaging, and will serve as the moderator for today's program. Dr. Perlin is chief scientific officer and senior vice president of the Center for Discovery and Innovation at Hackensack Meridian Health. And Professor Reddy is president of the Public Health Foundation of India. Over the next hour, they will discuss the current science on COVID-19 variants in the US and globally, and will examine the public health implications of COVID-19 variants in India and globally. And now, Dr. Mahajan, I'll hand things over to you to begin the program.
Thank you very much, Molly. Welcome to this seminar on evolution of COVID-19 variants, perspectives from India and the United States. Good morning and good evening to all the people who have joined us for today's session. And I believe it's already a few thousand from across the world and from different time zones. Thank you as we discuss the evolution of COVID-19 variants. I would like to extend a warm welcome to Dr. K. Srinath Reddy, President of the Public Health Foundation of India, and Dr. David Perlin, Chief Scientist Officer and Senior Vice President of the Center for Discovery and Innovation. Researchers and epidemiologists around the world have been predicting various models for potential spread of COVID-19 pandemic, which has really devastated many parts of the world in the last 18 months. We are facing a big challenge of continuous mutations of the COVID-19 virus, which makes it tougher to predict how the whole pandemic is going to unfold. Various models tell a different story about the loss of life in the coming months and a range of possibilities given the facts on the ground. In addition, while rates of infection and death have begun to decline in the United States, we have seen in the last 2 months devastating surges of the virus in India and several other countries, reinforcing the fact that there is need for global collaboration in order to contain and finally end the pandemic. While the future of the pandemic and its projection remain uncertain, though vaccination rates are increasing, yet we find that there are inequities even in vaccination across the world. Hence, it is still helpful to understand the most likely outcomes as well as the best and the worst case scenarios in order to guide local and national decisions that can lead us closer to the other side of the pandemic. It's wonderful to have you on board, and in the next 50 minutes or so, we will hear 2 experts from different parts of the globe talk.
Oh, it looks like Dr. Mahajan may have gotten a little bit disconnected. So why don't we move right into
A pleasure for me.
Oh, we can hear you now.
Invite Dr. David Perlin on.
So I think in view of some of the technical difficulties with Dr. Mahajan's presentation, I think I will then look to get started. If that's right, Molly, can we proceed?
Yes, please go ahead. Thank you.
Okay. So I want to thank Dr. Mahajan for the introduction to today's webinar. And what I was hoping to do as a prelude to Professor Reddy concluding with today's remarks is to talk about some of the science behind the evolution of the generation of SARS-CoV-2 variants of concern. What are those concerns? What should we be worrying about? What do we know about various variants in terms of transmission and their health effects? And then finally, what is the impact on potential therapies as well as on vaccines? So let's get into it. So let's start at the very beginning. We all know that with SARS-CoV-2, like other coronaviruses, often airborne, first interaction with the host or with us is through the respiratory pathway. And then as virus is taken in and interacts with the lung epithelia, it infects. So there's binding of the virus to the surface of the epithelial cells. It then gets brought into the cell. It then co-ops the host cell's replication machinery. It allows the virus to replicate. Virus then gets released, and then that triggers the immune response. And it's that very pronounced effect on the immune system that we see for respiratory failure. And then, of course, more and more cells get infected. Then you have destruction of lung tissue with effects throughout the body. But the key to the viral infection is, in fact, the binding of the virus to its receptor. And so what I've shown here, if you can see with this pointer, this is the host receptor, and the spike portions or these spiky protrusions from the virus, these are the spike proteins, and the spike proteins have a region which is called the receptor binding domain. That's the lock and key component of the virus, which is shown on the right here in this little portion. and the receptor on the host cell, which is the angiotensin 2 receptor, part of the angiotensin pathway. Now, we know that when the host responds and you form neutralizing antibodies, the antibodies that protect against virus are those that are directed against, largely against the receptor binding domain. So if you bind an antibody, now the virus is no longer able to interact with its host cell and can't replicate. Now, in terms of the development of virus variants, we recognize that RNA viruses, like SARS-CoV-2, rapidly generate mutations. And this, if you look here on the left-hand side, this is the mutation rate. And you can see where viruses stand. They have a very high mutation rate. This is negative 1 in 10 to the 5th. That means each time the organism replicates, it's creating another mutation. Whereas if you look at humans, it's very, very low. So we don't mutate quite as rapidly as viruses. Now, in terms of in terms of the difference, viruses are replicating, especially the RNA viruses, 1 million to 10 million times more rapidly than we do as humans. And within, for example, the SARS coronaviruses, if we looked at coronavirus 1 from 2003, which is shown here, it actually, its mutation rate is actually slightly higher than what we see with SARS-CoV-2, and yet it's still very, very high. And what this has done is, even from the very beginning of the pandemic, China and then in Europe and then throughout the rest of the world, that as virus variants arise, then you have these extensive mutations, then they lead to individual lineages, and you can see each of the arms here represent lineages. And if you look globally, then these lineages become entrenched in various regions within countries. But the virus is continuing and has been from the very beginning evolving quite rapidly. Now, each mutation has the potential to change viral properties for the next generation of viruses, especially when it's under selection pressure by the immune system. So antibodies that are being directed to the antibody binding domain, to the receptor binding domain, virus mutations that then alter that binding have one of three possibilities. It can leave the virus unchanged. It can lead to a decrease or loss of function. So the virus essentially disappears. And that's what we believe happened with SARS-1. It sort of mutated itself out of existence. Or you can get viruses that then have gain of function mutations, where they become more transmissible, more infectious. And those are the ones that we're worried about and the ones that we're beginning to see now. Okay, so if you look at this in terms of neutralizing antibodies, mutations in the spike receptor binding domain, they'll arise that they increase the binding of the virus to its receptor. So there's a competition between the antibody, the receptor binding domain, and the receptor binding domain of the virus and its host. So if you increase the interaction of the virus with its host, the antibodies don't bind quite as readily. And so the consequences of this are that the mutations can enhance the relative infection rates, it promotes more infection, and it reduces the potential effectiveness of vaccines or naturally occurring antibodies from infection, and may in fact facilitate reinfection, which is what we've seen with some variants. Okay, so if you look in the case of the UK variant, this is the so-called alpha variant, which was first identified in the United Kingdom. There were a number of viruses that were circulating, which became predominant, but what's shown in red is what we call the B117 virus. This is the one with higher transmission rates and began spreading throughout the United Kingdom. But indeed, as we look at other viral variants, whether we're looking at the alpha variant, or the beta or gamma variant, this is the the 351 or P1. Those two variants have a slightly different characteristic. They're highly transmissible, but because of this mutation, E484K, this is a glutamic acid, an amino acid change to lysine, a very significant change in the receptor binding domain that allows this particular strain to what we refer to it as an immune escape variant, meaning it's now not as sensitive to antibodies, to neutralizing antibodies. And so in the case of the P1 or the gamma, the gamma variant, it was seen in Brazil. It rapidly spread in South America, and we've seen it in North America and elsewhere. But we also see this particular signature mutation in variants for example, in our region in New York and New Jersey. And the B1526, which is prevalent in New York, was first identified in New York City and has become the predominant virus in New York City. It spread very, very rapidly in New York City and now in New Jersey. And so within a matter of a few months, it's sort of taken over the virus population. Now, it's really important to understand some of the characteristics of this. In fact, the alpha virus, the one originally identified in the UK, was somewhat less sensitive to neutralization by monoclonal antibodies. But more importantly, the E484K substitution that we see in New York, that's characteristic of the beta and gamma viruses, those then are much less sensitive to antibodies. And in fact, in some cases, some of the antibodies, like in the antibody cocktails, they're no longer effective against the virus. And you can see this here. This is from David Ho's work that was published this past February in Nature. And what it's a complicated slide, but just to give you the gist of it, we look at the strength of the antibody binding to virus. And what you see is wild type, that's what we would call this, quote, normal virus, E484K. And if you look here on the right-hand panel of this antibody, there are several antibodies that completely drop out. They're no longer neutralized. The virus is no longer neutralized by those antibodies. So, for example, the Lilly antibody cocktail, which many hospitals, certainly in the US and around the world, were using as first-line therapy, that cocktail was no longer effective against this variant, whereas some of the others were. And then if you look at vaccines, either from vaccination, sera from vaccination from patients, you can see there's a diminution of the response of the antibodies from vaccinated patients with this particular variant. Same thing with somewhat with convalescent plasma from patients. So certainly this particular mutation, this E484K, is particularly troublesome as it does in fact confer a level of immune escape. Now, we've seen in our region, it's a very good example of how rapidly these variants can take hold. We began to see the alpha variant, the characteristic mutation, this 501Y, and then the 484 escape. And you can see back in December, we didn't have any viruses, or we had one small outbreak, but essentially we had very few viruses in our region. In January, it was a few percent. By February, we were beginning to see both. By March, April, and now May, more than 60% of the viruses in our region are the alpha virus, and they're the alpha. And then we're beginning to see the alpha virus with immune escape viruses that are coming online. So they're both together. So we have high transmission and we have immune escape, which is clearly troubling and makes things more problematic. Nationally and in the US, this is data from the Centers for Disease Control and Prevention. You can see how from March now to May, the alpha variant has become the predominant variant in the US, although other regional variants in California, New York, and other parts of the country, in Texas and Florida, are also relevant. Now, the World Health Organization has indeed named these the alpha variant, which is the one originally in the UK, B117. Beta, these are variants of concern and so forth. We'll talk about Delta in a few minutes. But the variants of concern increase transmissibility, and they have a detrimental change in the epidemiology. You can see how fast they sort of take over. Increase in virulence, they can cause disease more rapidly, and in many cases, enhanced hospitalization. And then, of course, decreased effectiveness of public health measures, as you have viruses that will infect more readily than they're able to transmit, and they have implications, obviously, for both our diagnostics, vaccines, and therapies. There's variants of interest, which are also important, and we need to keep track of them. But they're at a somewhat lower level. They may still have immune escape properties, but they're not necessarily as highly transmissible, and we don't see them as broadly in the community. They tend to be more regional, but significant nonetheless. And then you can see that we've had this global spread of specific lineages, whether it's the alpha, beta, or gamma, or others throughout the world, and now the Delta strain, which I'll talk about now. So the Delta strain, which was originally first reported in India back in December, has spread to now at least 62 countries, probably more. And it's designated as the B1617.2. This is a family of viruses, and this is the one that we're most concerned about within that family of strains. And it's deemed to be 60% more transmissible than the alpha strain. That's the one that was first identified in the UK, and that immediately tells you how rapidly this strain can spread. And indeed, if you look on the bottom half here, in India, it's become the predominant strain. Now in the United Kingdom, it has, and the US is reported to be 6%, but that's increasing as well. And if you just look at the example of Delta in the United Kingdom, here it is in May 15th, where just under 39% of the viruses reported were of the Delta variety. Fast forward 2 weeks, and now more than 90% of the viruses in the UK are being reported as Delta, and clearly very troubling, but it just shows how rapidly these viruses can transmit. The key to the Delta variant is here. This is the 617.2. It has these characteristic mutations, then the L452, this is leucine to arginine, and this threonine to lysine at 478. These are the two key mutations. They're within the receptor binding domain. As I pointed out, that's how these viruses are evolving. They want to maximize their interactions with the host. So what is the impact on vaccination? Well, it's variable. Let's look at some of them. This is data from Pfizer and the BioNTech. This is one of the mRNA vaccines, and this is a study that was done in Qatar, where they have a high prevalence of the UK and South Africa. That would be the alpha and beta variants, and these were they showed really strong efficacy, 90%. This is with the alpha variant, and a little bit less, about 75%. With the beta, the 1351, that's very good. 75% is an outstanding level of protection. So these viruses, although they're a little less sensitive, as I indicated before, they're still highly protective. The vaccines are still highly protective. The Johnson & Johnson vaccine. This is the vector vaccine, had very good, very strong responses in trials done in Brazil and South Africa. Interestingly enough, they had 88%, 82% protection. It's believed that protection is both antibody mediated, so neutralizing antibodies, as well as the other component of the immune system, and that's T cell mediated protection. We won't get into that very much, but this is another important component. But there was an interesting finding, and that is that individuals who had who were infected with these variants produced 5 times fewer neutralizing antibodies. Now you're dealing with logarithmic levels, so you're still having really strong responses, but nevertheless, the virus did induce a reduction in antibody production. If you look at the AstraZeneca Oxford vaccine, it did not do as well with the beta variant in South Africa. There was essentially no difference in transmission with or without vaccine, or was rather modest at best. However, I would point out that despite the fact that, and this led to public health measures in South Africa about the use of this particular vaccine, nevertheless, it is important to point out that they were looking at mild to moderate cases of disease, and in fact, even with the J&J vaccine, that cases of severe hospitalization were not observed. So the vaccine was in fact still effective, just didn't prevent some of the mild to moderate cases. In terms of other vaccines, the Covax and Covishield, which are being used widely in India, they also induce fewer antibodies, yet were effective against, this is now the Delta variant, and Pfizer, the Pfizer vaccine, the mRNA vaccine, also produced less antibodies against the Delta vaccine, yet it was still also quite effective. So if you put this in tabular form, this is just some of the more prominent vaccines. Of course, there are many more, and you can see the relative effect on the major variants of concern, alpha, beta, gamma, delta, and which vaccines were either most likely to be effective, yet in many cases we need more data, and some with the existing data are probably not so effective. I would point out that this is a study that we had performed with David Ho's group at Columbia. This is from an outbreak that we had in New Jersey. And these are individual patients. These are serum titer levels. And you're doing dilutions of serum titer. The hash line indicates what we call percent neutralization. This is level of protection. And so all of these patients were infected with an E484K variant, and they all recovered. They mounted antibody responses, which were cross-neutralizing at a low level to all the known variants of concern. However, if you vaccinated, which is shown here in these circled levels, you can see you can boost the level of antibody 10 to 50 fold. And when you do that, you now get cross protection against essentially every known variant, except perhaps MERS, which is not relevant with respect to COVID-19. Nevertheless, it illustrates vaccination, even people who've had recovered from variants is really important to bolster the immune system and induce long-lasting protection. And finally, in this variance study, what we can say is that, that we have a range of virus attributes and alpha, beta, gamma, epsilon, delta, and so forth, high transmissibility of delta, immune evasiveness. Beta, has immune evasiveness, not as highly transmissible, whereas alpha is, and the combination of these make for dangerous variants that are highly transmissible, immune escape, and that's what we're concerned about, and that's why we need continued surveillance and monitoring of what's happening in this pandemic. It's rapidly moving and it's not going to change. So finally, I'll conclude by saying virus evolution has been an important component of the COVID-19 pandemic. Variants of high concern, high global concern, such as strain Delta, show enhanced transmission and ability to cause disease. Some variants may partially impede antibody development and can be controlled, though, effectively by existing vaccines. The best way to control variants of concerns is to reduce the reservoir of virus by broad vaccination, and that has to be our goal. So I thank you very much, and I'll take questions. Well, actually, I think.
Thank you very much, Dr. Perlin. We will be taking the questions at the end. May I now invite Professor Srinath Reddy, and he will be talking about his experience of the public health implications of COVID-19 variants in India as well as globally. Professor Reddy, please.
Thank you very much, Dr. Mahajan. I think Dr. Perlin has given an excellent exposition of the science of virology and immunology as it pertains to SARS-CoV-2 virus and its variants. And he has elegantly explained how the variants have arisen, how they are classified, what their potential for influencing infectivity as well as possibly vaccine escape is. I will not dwell into the science of virology and immunology, but will try and deal mostly with the public health perspective, but with a little throwback to evolutionary biology as an additional perspective. Indeed, when variants do come up, one of the biggest questions that we ask ourselves, particularly when we are looking at what implications it has for the public health response and the health system response is it more infectious than the wild virus or any other variant that preceded it? Is it more virulent? Is it more capable of vaccine evasion or escape? And also, is it less responsive to therapeutic interventions like monoclonal antibodies or antiviral drugs? And while we are trying to answer that, we have to also understand how so many variants are arising in different parts of the world, and of course, transmitting themselves with great ease to other parts of the world. We have generally tended to look at viruses and variants in a very linear fashion. But viruses are complex adaptive systems. Indeed, there is a book by that very title, a book by Ricard Soleil and Santiago Elona, published in 2019. And it is very clear that they must find ways to become more infectious while escaping the effects of immunity. And that is their evolutionary biology pursuit. Joshua Lederberg, who for microbial genetics, particularly bacterial genetics, got the Nobel Prize in 1958 and is considered to be the father of microbial genetics, wrote a seminal paper in Science in 2000, in which he said, it is our wits versus their genes. So we have to use our wits to try and overcome what their inevitable attempt to transform themselves into new forms through their genetic innovations. And that is not always an easy battle, because the viruses can mutate much faster than we can produce vaccines. Therefore, we have to combine a number of public health measures to contain the transmission and to limit the course to mutation, even as we try and pursue our quest for vaccines and effective drugs. Evolutionary biology, therefore, must provide access and to new information. And what the viruses seek is access and staying power in the primary host, and to seek shelter and survive in a reservoir host. So we first need to identify what the primary host is, what the reservoir host is, and what a secondary host might be, and try and interrupt the process of transmission in each of those links. In this process of balancing between survival and speed, for the virus, there is usually a trade-off between infectivity and virulence. And if we can actually stall the virus from easy transmission, there is a theoretical possibility that it will try and become more infectious but less virulent over time, because it has to continue to propagate its own species. Therefore, public health comes in as a very important measure in order to slow down the transmission of the virus and try and limit its opportunity not only to spread fast, but also to mutate into more virulent forms and possibly nudge it to become less virulent over a period of time. But it may not always be easy if there is an abundant host available. Different variants arising in different places are also a matter of interest. And it's not purely serendipity that they're arising in so many different places with different characteristics, but many of them having similar features. So some of the characteristics are shared and there are some differences. Therefore, there is some self-similarity in nature. And to understand this, perhaps we'll have to go to the theory of chaos and fractals and look at what are called the Mandelbrot sets, which were described by a mathematician, Benoit Mandelbrot. And then we can find out there are different points of origin, and each proceeding towards infinity with exponential growth. And therefore, it is our business, our necessity, our imperative to try and stall that exponential growth of these different foci that arise at different locations. And that is why we are seeing different variants arising in different places, growing exponentially initially, but we are succeeding to some extent in stopping their growth further and limiting their exponential growth through our public health measures, which includes vaccination, of course. What are the conditions that create variants? Clearly, a lengthy stay in the human host, in a human body, gives the virus the luxury of time to experiment and mutate, to try and acquire characteristics which might give it much greater infectivity or virulence or both. And such a host is available in an immunocompromised person. In fact, the alpha variant in the United Kingdom was supposed to have arisen in an immunocompromised patient who was ill for a long time and perhaps gave the virus an opportunity to mutate at leisure. It can also happen in an elderly, relatively debilitated person where the immunity is at a low ebb, and again, the virus can either kill fast or stay in the body for a long time to mutate. And persons on ventilators for a long time are obviously going to give the virus a greater opportunity to stay in the human body and again play with the idea of creating newer mutations to change its character and its capacity to transmit. Therefore, the slow pace of vaccination becomes a challenge. When we actually have very slow vaccination and the virus is able to transmit easily across many people, and some of them will harbor the virus for a long time in their body, then the opportunity for mutation increases. And the faster we vaccinate, the wider we vaccinate, the less opportunity we provide the virus to mutate. Unfortunately, in some of the places where the virus has actually mutated and where it has arisen in the first place, or at least identified in the first place, like Brazil, South Africa, and India, the vaccination rates have been rather slow, at least at the time the variants first were identified. But then there have been also places where vaccination trials have been going on, and fairly energetically, like the United Kingdom and the United States of America. Before a certain population threshold of vaccination was crossed, even the fact that the vaccines were trying to limit the capacity of the virus to transmit could have possibly induced mutations as a response. So there are very many ways in which our encounter with the virus in the battlefield of public health can actually result either because of very slow vaccination or in the initial phases of the vaccination trials, possibly give an impetus for the viruses to develop into variants with mutations. So what we really see is that the variants are attempting to enhance their armory of mutations in order to overcome what we are throwing at it. And therefore, our capacity to throw our armory at them with greater speed and accuracy is going to be absolutely critical. And now we are seeing alpha, beta, gamma, delta, all of the variants that have been described have come up now in different parts of the world, and some of them are replacing the others and becoming more dominant. We have just been introduced to a new variant again, the AY.1, or what's been called the B1617.2.1, which has been identified for the first time in Europe, but is now again, and it was unveiled to the world. It was identified in March, but unveiled to the world only a couple of days ago. It has also been spotted in India and some other countries. And interestingly, this particular mutation has an additional mutation called K417N, which it has picked up from the beta variant, which was initially identified in South Africa. And therefore, this additional mutation now confers even greater infectivity on it. Therefore, there is a race between us and the virus, the race between the vaccines and the virus, and therefore we need a composite set of public health responses. And what are they? Firstly, we must recognize that whether it is the wild virus or any variant, there are only three portals of entry by which they enter the human body: the nose, the mouth, and the eyes. And if we can close those gateways as effectively as possible, then we can actually prevent the virus from getting easy access to human bodies and replicating itself or even acquiring additional mutations. So what we need to do is to reduce the transmission by barricading the portals of entry into the human body and creating a bumpy road for the virus to travel. And we now have the knowledge and the instruments to do that. We know that masks are very effective. We know that ventilation with free flow of air doesn't form the viral clouds. It doesn't allow the viral clouds to form and accumulate. And therefore, well-ventilated spaces are protective. We also know that we must avoid crowds to prevent super spreader events. And therefore, preventing indoor or outdoor crowds and ensuring that there are no super spreader events is going to be absolutely critical till we can actually be sure that our vaccination rates at the population level are picked up to the extent that the virus is no longer such a threat. And we know that it can happen. Like, for example, in Brazil, in Serrana, it was recently reported that when the vaccination mark crossed 60%, the population, the deaths came down by 95% and infections came down by 86%. similarly, on the 1st of June, when the UK crossed the 60% vaccination mark, they had recorded 0 deaths for the first time since March 2020. So we do need a higher level of population vaccination, but till that time, we have to hold the fort by all methods of preventing the transmission from person to person. And we do need to increase the pace of vaccination, but we also now need to ensure that our vaccine science itself is responding to the challenge of the variants. If the variant is throwing many more mutations at us and is trying to evade or escape from the vaccine, then we need to modify our vaccines as well. Fortunately, so far the vaccines we have seem to be working well, but we have to make sure that we do have additional vaccines or modified vaccines in case some of the mutations begin to have greater levels of vaccine escape or completely evade the vaccine-induced suppression. And how do we do this? We do this by tweaking existing vaccines, and that's already being done with mRNA vaccines, where some of the modifications are being made to Moderna as well as the Pfizer-BioNTech in order to try and address the variations and the mutations. And we also may have to invoke and depend more on the T-cell immunity, which acts on intracellular virus presence, whereas the antibodies produced by humoral immunity or the B cells act only in the bloodstream on the surface of the virus by attacking the spike protein, but they do not reach out to an intracellular virus, but the T cells can. Therefore, we may have to have vaccines which can have a much stronger T cell response. We also may require a broader band of immunity to other antigens rather than targeting only the spike protein. In fact, in a very nice recent review by Catherine Wu in The Atlantic, she says we may have to go beyond our monogamous relationship with the spike protein and look at other elements of the virus as well. So we may need new vaccine pathways, particularly those packed with additional bits of the virus, that means which offer a larger platter of antigens for the body's immune response. We need more, we need to have our immune system prepare for more targets to attack in the virus. And we need to loop in untapped reservoirs of immune cells that have no interest in the spike protein, but can attack other parts of the virus. Neutralization still will remain very important because the spike protein will probably stay as one of the key areas for attack for us to target. However, we may need additional areas of the virus also to attack. And we have seen some of the vaccines are using whole virus particles, including some of the inactivated virus vaccine that has been prepared in India and is currently being used. Therefore, if you have a larger platter of antigens, the broadband immune response may actually act against other parts of the virus. And even if the virus tends to escape, the immunity that has been directed at the spike protein, still the other areas can possibly be subjected to the attack. But we have recently also seen that in California-based Gritstone Bio, a whole package of antigens has come in with one surface protein, the spike protein, being not only being targeted, but also two interior proteins, that is the nucleocapsid, which helps the virus to package its genetic material, and ORF3A, which helps the newly formed viruses to move out of the cells of the human host. So you have a whole package of antigens which are being targeted. So we are now seeing that neutralizing antibodies, which only latch onto the outside of the cell, are like cameras, as Kathleen Wu puts it. But the T cells can recognize and kill infected cells, intracellular viruses. They are more like X-rays, as Dr. Mahajan might well appreciate. They actually have a much better vision. So we do need some of the non-spike protein immune targets, because these actually mutate also more slowly in the virus than the spike protein. This raises hopes of immunity, which is both potent and long-lived. And T cells are also not presently vulnerable to vaccine escape. So we do need a greater panoply of vaccines T-cell dependent vaccines may be particularly useful in immunosuppressed persons who are on drugs that blunt the ability of B cells to produce antibodies. So those who are immunosuppressed because of cancer chemotherapy or others may particularly benefit from vaccines which can really potentiate the T-cell response. Therefore, to sum up, our response in terms of public health has to be to protect ourselves, to prevent the virus an easy entry into our body, to obstruct the virus in terms of its transmission by not allowing superspreader events, vaccinate fast and wide, and develop new vaccines with innovations which can match the variations in the virus. Thank you.
Thank you very much, Professor Reddy, for giving us insights into the public health part and also the vaccination. Now we are left with about 8 minutes for questions, and we have a lot of questions that have come in. So I would request Dr. Perlin and Dr. Reddy to keep the answers short. My first question is from Madhav Kulkarni, who says, what are the factors that promote and speed up mutations? Dr. Perlin, please.
So the mutation rate is sort of set by the virus. It will continue to mutate. The key factor here is what is the selection pressure, and the key selection pressure is going to be it will be the immune system. As we vaccinate more broadly, it will be the immune response that's invoked by those vaccines, in particular neutralizing antibodies. So as as Professor Reddy said, the longer virus persists in the host, the greater the opportunity there is for the development of viruses that can escape the immune system.
Thank you, Dr. Perlin. The next question is for Dr. Reddy from Laura Marine. How is the data of people infected with the different variants obtained? Are specific COVID-19 tests available for the population? Common tests only show positive or negative results. So how do we go about knowing what is what, Dr. Reddy?
Well, of those who have tested positive for the virus, and particularly on an RT-PCR test, at least 5% of the samples should be subjected to genomic analysis, and that requires special laboratory facilities. And we have seen some countries have gone up above 10%. In fact, a very large proportion of the samples in Britain are now being subjected, even more than half are being subjected to genomic analysis. And some countries are well over 10, 15%. Therefore, the more samples of the virus positive tests that we can actually subject to genomic analysis, and the faster we do it, the more likely we are to pick up the variants. And the earlier we pick up the variants, sooner we can actually try and stall their transmission.
Yeah, I'll just comment as well that within our network, which covers most of New Jersey, every positive virus gets sent to us. We then analyze it through a high throughput genetic analysis where we can look at several hundred viruses a day and look for those specific mutations that are relevant to the variants. And then if we want, then do whole genome sequencing, which is a much slower but more extensive analysis. But we agree that at least 10% of the viruses we want to look at, and in many cases, we are in fact in our region very close to 50% of analysis.
That's great. The next question is from Arvind S., who writes, I've read that viruses ultimately become less virulent over time. Is that only when it is not able to infect people anymore and the strains that let the host live and coexist have greater chance of propagating? Can the virus hang around in the body for longer than 2 weeks? Dr. Perlin, would you like to take that?
It absolutely can. I mean, we have we have a number of patients with persistent, what we more or less would call viral colonization. The reality is the virus needs the host, it needs the humans to persist. So if it doesn't kill the host, then it'll persist. Ultimately, it should get to a state where it doesn't want to kill and cause disease, but it wants to continue to replicate. And so fundamentally, we do, and we have seen a number of patients who have persistent viral colonization.
Thank you. Thank you. Dr. Reddy, has there been any experience with combination of vaccination types? If so, are there any added advantages?
Interestingly, recently, a lot of information is coming on this. Firstly, even when you look at the virus vector vaccine, the Russian vaccine, for example, they use two different human adenoviruses in the first shot and the second shot, so that the so-called innocuous virus vector itself does not produce immunity against itself for the second shot and therefore become less capable of delivering the package it has to carry of the DNA code for the spike protein. So the postman itself should not get hurt in the process. But also now, some trials have shown, for example, in Germany, that using the AstraZeneca and then the Pfizer-BioNTech actually produce 3.9 times more, a better immune response than a single vaccine type alone. And there are other experiments being tried out now of trying to look at different vaccines in combination. And perhaps some of the vaccines can stimulate the T cells better than just the antibodies. So I think we will get a lot more information over the course of the year about this mixed vaccine combinations.
Thank you. Dr. Perlin, will all of the vaccines we are currently getting have to be updated yearly, like in the case of influenza? What are your thoughts on that?
Well, we'll have to see how effective the current vaccines are in terms of sustained immunity. I think we as a network, we're starting to monitor this. I know many others are looking at antibody levels, looking at levels of protection. I think it's best for us to prepare that, in fact, we may have to update the vaccines. I know the manufacturers are, in fact, looking at that, Pfizer and others, that this is something they're preparing for, anticipating that the virus will continue to evolve as we see, for example, with the Delta variant, other immune escape variants. So I think, to Professor Reddy's earlier point, I think that the manufacturers also need to be smart about this. We need to begin to focus on other parts of the virus, which are less mutable, and take the approach that we're trying to use with influenza.
Thank you. We have just about a minute left, and this will be the last question. Dr. Reddy, if you could take it. And it says, yes, this is Kenneth Onuoha, who asks, what do you think might be primarily responsible for the widespread of the COVID virus in India? Is vaccine hesitancy a major concern in India, or is it more of a low availability of the vaccines? 30 seconds, sir.
Well, right now there is a challenge of lower availability, but at least in urban areas, there is no vaccine hesitancy. In fact, now there is a certain sense of vaccine urgency. In rural areas, perhaps we need to overcome some of the vaccine hesitancy, but the vaccine supply also has to increase simultaneously.
Thank you very much. I must say we've had a great interactive session, and it's time for me now to hand you over to Molly Lowe. Over to you, Molly.
Thank you so much, Dr. Mahajan, and to all of our speakers.
This concludes today's webinar, which I would like to emphasize. We are so happy that this was a joint initiative between the American College of Healthcare Executives and NATHEALTH, made possible by that joint initiative and collaboration. So thank you for attending. Drs. Mahajan, Dr. Reddy, and Dr. Perlin, I'd like to thank all of you for your collaboration and for sharing your knowledge and insights into the various COVID-19 variants and the implications of these variants on hospital and health system decision-making at an international level. In closing, we also thank each of you for what you are doing every day to support our communities and help us navigate during the ongoing pandemic. We're so grateful for all that you've done this past year and continue to do. And as a final reminder, watch for the follow-up tomorrow with links to the session evaluation form, the recording, and updated version of the handout. Thank you for joining us today, and please enjoy the rest of your day.
Thank you.